Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and what we were discussing about the different chromatography techniques which people are using to purify the protein or the other factors from the cells where they are overexpressing these components. Now, how to perform this affinity chromatography in your laboratory? Let's take so for this I would like to take you to my laboratory, and where we are going to show you each and every step how to do the charging, how to do equilibrate the columns, and then how to purify the protein using the nickel NTA affinity chromatography.
For protein purification, first we have to inoculate the culture into this uh, larger volume of ELP class. Then we will induce it. So first I will show you how to inoculate. This is the single colony grown overnight culture. So we can use for the inoculating into large cultures. So this process should be done in aseptic condition. So that means we have to use laminar airflow for this purpose. So and also we have to remember we should include suitable resistant marker like ampicillin or kanamycin, this kind of antibiotics, or this is depends upon what vector, what resistant vector you are having. So in this case we are using ampicillin as a antibiotic. So let's start. uh, we have lysed the cells using sonication. Now we have to centrifuge the lysate to get supernatant. So that supernatant we load on to nickel NTA column and purify the protein. So I will transfer into 50 ml centrifuge tube, then I will centrifuge. While the centrifugation is going on, we have to wash the column using first, this is in 20% ethanol. So we have to wash first with water, then equilibration buffer. So let it drain completely, that 20% ethanol. Then we will add water, double distilled water. So at least 5 column volumes of water should be added to remove completely. And next we will equilibrate with the lysis buffer, the buffer which we used for the lysis of the bacterial cells. Before equilibration of the column, we have to charge the column. Nickel NTA, there are two types of bits are there. One is already readily charged bits which comes from company, and another one is we have to charge. They will give only in NTA agarose bits. So here what we will do is we will charge the beads with the nickel, and then we will equilibrate. We already washed the column with water and 0.2 normal NaOH again with water. So now we will equilibrate. So this is a nickel hexachloride solution. Uh, So we will keep in this condition at least 20 minutes to charge the beads. After that, we will remove nickel NTA. We will elute the nickel solution, and then equilibrate with the lysis buffer. So after 20 minutes we evaluate the nickel solution. Uh, Next we will equilibrate with the lysis buffer. We have to wash at least 2 column volumes to remove any free nickel which exists in the beads. So after equilibration, next step we will load the lysate and then incubate for binding. This is a paraffin. I am going to close this and open the pan. I'm going to keep it here. So once column packing is over, we kept it in ice and we'll keep in this condition for at least 2 hours for binding. So that histone protein will bind to the nickel NDA, and in further steps we will elute the protein. After incubation with beads, we have to follow another 3 steps to get completely purification done. First step is we have to wash with the equilibration buffer. First, after the beads taken out from ice, you have to remove the outlet so that all the flow through other than beads will be taken out. And the next step is we have to wash with the equilibration buffer. And the third step is we have to elute the sample, elute the protein, histone protein using imidazole containing buffer. For all these buffers, the pH should be adjusted prior hand. Not like you have to first you take the buffer, lysis buffer, and you have to add imidazole. It's not like that. It may increase the pH of the buffer. So after compiling all the lysis buffer with the imidazole, then we have to adjust the pH so that throughout the procedure the pH won't change. So this is a flow through. Whatever we are getting is flow through. In next step we will wash with the lysis buffer. In this step we are going to wash with the lysis buffer or equilibration buffer. So this is the lysis buffer. Before doing this, we have to observe the beads. We should not directly load onto beads. You just have to pour through corner, through the wall of the column. Otherwise, it may disturb the beads. So protein may also degrade. So this we have to keep in mind while doing this washing. While doing purification, we have to remember that every time you are introducing new buffer, that time you have to collect the fraction, and this can be used for the running SDs and testing the purity of the samples. And also the flow through part and the washing part, what we have collected, we have to keep it safely. After verification of the gel, only we have to throw. Say you are getting only 10% of the protein in your purified fractions and 90% of the protein eluting in the flow through. That time you can reuse the flow through for purification again, purifying the protein again. So you have to collect the fractions in a small microcentrifuge tubes. and we have to save those fractions. Name it and save. So we washed with the equilibration buffer and we also collected the flow through. Now it's time we will wash with the 20 millimole of imidazole. So this will remove any non-specific proteins binding to the beads. So we will wash with the 20 millimole imidazole containing buffer. Then we will subsequently elute in 250 millimolar imidazole containing buffer. In the final step, we are going to elute with the 250 millimole imidazole containing buffer. So what we are going to do is we have to incubate beads with this buffer for some time and collect the fractions. Now we have to collect the fractions. After eluting the complete fractions, we have to wash the column with water, then 0.2 normal sodium hydroxide solution, then again water. After final wash with the water, then we have to store the beads in 20% ethanol. So I will wash it and store it in the ethanol. While the washing is going on, we have to take 50 microliter of each fractions and run it on SDS phase. That will give the purity of the fractions. We have to heat the samples before loading onto SDS phase, and also we have to keep all these fractions what we have collected at 4 degrees Celsius for further confirmation of the purity. Once the purity is confirmed, we have to dialyze those fractions against our buffer of interest, then use for the further studies. So we purified the protein using nickel NDA column. We run the gel and stained or de-stained. So now it's time to document the gel. So we have to identify whether we got any single band fraction or not. So this is the gel I kept on white tray. Now just close it. So we have loaded marker and this from this side. Second one is the load. This is flow through, wash 1, wash 2, and these fractions are eluted fractions 1, 2, 3, 4, 5 serially. So as we can see, the eluted fraction showing a band corresponding to this protein, but the molecular weight can be calculated using the software, ImageLab software. So as we can see, in the protein corresponding to this purification his tagged one, it is going most of the fraction in the flow through. So we can use as I said in the video earlier, we can use this flow through action again for purification of the protein. You can incubate this flow through with the same beads, and you can repurify again, so that will increase the productivity getting the protein. So these are all other bands whatever we are seeing in the protein, this eluted bands, those are because of the contaminants or degraded protein. Contaminants sometimes may come because of histidine, 3 or 4 histidine having in folded state, that will give possibility to bind to nickel NTA column. And also washing, vigorous washing should be done. If you don't wash properly with a high amount of imidazole, that will give you this kind of non-specific binding. So with this we'll conclude the video. So I hope it will help you to help you in your work for during protein purification, or help you to understand how protein purification works.
Let's move on to the application part of the nickel to the affinity chromatography. So, affinity chromatography is a very robust technique or robust chromatography techniques which can be used for multiple applications. One of the application is that you can use the affinity chromatography to isolate the protein complexes. If you remember in our earlier lectures, we have discussed that suppose you stimulate a mammalian cell with the insulin, what will happen is that insulin is go and bind to the insulin receptor, and in this process the insulin receptor is going to get phosphorylated. Once the insulin receptor is getting phosphorylated, it brings the binding site for many of the adapter proteins. Once these adapter protein goes and binds to the insulin receptor, they actually relay this phosphorylation signal, and that is how they actually give this signal to the nucleus, and that is how the gene expression profiling is going to be changed, and that actually is going to change the overall physiology of this particular protein. So, suppose you are working in your laboratory and you are exploring this problem or the similar problem where you are stimulating a cell and you are looking for that what are the proteins are binding to this receptor, or what are the proteins are coming together to make a multimeric complexes, like some of the signalosomes which are being formed when you are going through when a B cell or T cell is under the active immune responses, like against the cancer cells or against the infectious organism. And if you are exploring any such problems, then you could be able to use the affinity chromatography to isolate these bigger complexes or multimeric complexes, and you could be able to identify each and every protein, and you could be able to answer many of the biological questions. What you are supposed to do is, so affinity chromatography can be used to study or isolate interacting partner of a particular protein, which means in this particular problem you are going to have one protein which is going to have the affinity for the matrix. In this case, suppose this protein is insulin receptor, because we are stimulating with the insulin. So, our protein number 1 is the insulin receptor. Now, insulin receptor, you are going to put some kind of affinity tag. For example, I have put the histidine tag, or I have put the GST tag, so that it should have the exclusive affinity for the beats. Now, what we have in this approach, the matrix is incubated with the protein number 1, which means the insulin receptor, and then you wash to ensure the tight binding, which means you ensure that the beads are having the insulin receptor present on its surface. So, what you have done, you have taken a column which has the affinity for the protein number 1. In this case, this is the insulin receptor. So, what will happen is the protein will go and bind to the beads which are present in the column. Then you wash to ensure that there is no non-specific protein present. So, now your protein number 1 is bound. Now, you take the protein number 2, or you can take the cell lysate, whichever you would like to. If you would like to study the interaction between protein 1 and 2, then you can add the pure protein 2 and you could ask this question, or if you would like to flow the pure lysate, you can flow the pure lysate. So, what will happen is, if you flow the pure lysate, the lysate protein will interact with the beads. So, the protein 2 has the option of interacting with the matrix or interacting with the matrix bound protein 1, means it is interacting with the protein number 1 which is bound to the matrix, or it has an option that it interacts with the matrix directly. If this is the case, this protein is not going to give you the answer. So, if the protein 2 does not have any affinity for the matrix, suppose you change the condition in such a way that the protein 2 does not have direct affinity to the matrix, but it will bind only if it is having the affinity for protein number 1, then in that case the protein 2 will be bound to the column only in a one condition, that the protein 2 has an affinity for protein 1 which is bound to the matrix. Now, you wash so that the non-specific protein which are binding directly to the matrix are going to be removed, and then you do the elution of this with the salt. So, what will happen is both the factors, the protein A, protein 1, protein 2 are going to be eluted, and then you are going to test these protein 1 and protein 2 onto the SDS page, and the pattern of the elution is going to tell you that both the proteins are interacting or not, or whether, if suppose you are using the cell lysate, then the multiple proteins are going to come out. In that case, you have to do the control experiments where you are going to incubate the lysate to the beads without containing the protein 1, and that actually is going to work as a negative control. So, that you will know that okay, these are the 10 factors which are binding to the matrix directly. Now, the eluted protein is analyzed in the SDS page or the SDS page followed by the Western blotting to detect protein 1 or protein 2. As a control, the cell lysate or the protein 2 is also added to the matrix without protein 1 to rule out the possibility of protein 2 binding directly to the matrix. So, the control is very much important, that you should be able to know that these are the lysate protein which are directly binding to the beads, not to the protein 1. In a alternate experiment, you can use the affinity chromatography to generate or to design a protease assay. So, in a typical protease assay, what you are going to do is you are going to have a peptide, then you put a affinity tag onto this peptide, and then you use the affinity beads. So, what will happen is this is going to be your sample. So, you can change the peptide, you can have the complete peptide library, you put it into the 24 well dish. So, in this 24 well dish is going to have the different peptide sequences which are bound to the beads in separate wells. Now, what you are going to do is you treat them with the protease, and what will happen? The protease is going to cut the peptide at this point, okay, which means it is going to keep the tag bind to the beads, and the remaining peptide portion is going to be eluted. This peptide you can collect into the supernatant from all the beads, all the wells, and then you can perform the MALDI analysis, which means you can collect these peptides and do a mass analysis, and depending on this mass profile you could be able to deduce the sequences or deduce the places where the proteases is cutting in each well, and depending on this analysis you will be able to determine the protease cutting site of that particular protease. The fourth is you can actually be able to do the immunopurification, which means you could be able to purify the cells of your interest. Okay. This is important in terms of the places where you could be able to suppose you would like to purify the bone marrow cells, or suppose you want to purify the stem cells and so on. So, in those cases what you do is you use the some affinity molecules which will going to have the exclusive affinity for that particular immune cells. So, the evident biotin system is used to capture and isolate cytokines from the immune cells. Biotinylation of antibodies allow immobilization of antibodies in the correct orientation on the streptomycin coded glass beads. Lymphocyte lysate is passed to the column packed with the glass beads containing antibodies by cytokines. The cytokines are then eluted by the flowing buffer of decreasing pH or by cheotropic ions, and the antibodies remain bound to the column due to strong affinity between avidin biotin, which is resistant to this chemical treatment. So, in this apart from isolating the cells, you can also study the cytokine profiling from these cells. So, every cytokine is going to have its antibody. So, you can use that antibody coupled to the avidin and biotin system. So, avidin is having a very strong affinity for the biotin, and what will happen is that you can take the this lymphocyte lysate and pass through to the column, which actually contains this particular kind of antibody. Then you wash and elute with the ligand. So, what will happen is when you do the elution, only those cytokines will come which are been having the affinity for the antibodies which are present into these beads, and that is how you could be able to identify the cytokines which are being eluted or which are being secreted from these lymphocytes. So, this is all about the different chromatography techniques which you can use to purify the factors from the cells overexpressing your protein or other metabolites. Now, let us recap and summarize, and also should understand how you can exploit these different chromatography techniques to perform the protein purification or perform the purification of your factors. So, what we have discussed about the ion exchange chromatography, which is going to exploit the charge which is present onto the protein. Then we have talked about hydrophobic interaction chromatography, which is going to be exploit the hydrophobic patches which are present on the protein. Then we have discussed about the affinity chromatography, which is going to work on the exclusive affinity between the receptor and ligand, and then we have discussed about the gel filtration chromatography, which is going to work on the surface area of a particular protein, and surface area is directly proportional to the molecular weight of that particular protein, as long as we are considering that all these proteins are of globular in nature. Now, let us see how you are going to plan out the protein purification, or how you are going to plan out the purification of these factor from the cells which are overexpressing. So, what you have is you have started with the cells. The first is you are going to broken these cells, or you are going to destroy the cellular integrity, that actually is going to give you the cell lysate. Now, depending on the type of purification strategies you are going to use. So, suppose the factor what I am overexpressing is containing a tag, then in that case I will use I will use the affinity chromatography. But imagine that I do not have any such thing and I would like to use the conventional chromatography. In that case, what I will do is, first I will do a ammonium sulfate precipitation. So, what I will do is I will use the different concentration of ammonium sulfate, which means from 0 to 100%, and if you have not recount, you can actually go through with the Google or some other sources, and that will actually tell you that what amount of ammonium sulfate you have to add into a cell lysate to get the 30% saturation. So, the 100% saturation is the maximum what you are going to get. So, and that is actually theoretically you may not get the 100% saturation in many of the cases, but what you can do is the first step you can do is you can do ammonium sulphate precipitation, and that actually is going to give you the different fractions. All these different fractions can be collected and can be tested for the presence of your protein. Either it will be by Western blotting, or it would be by the enzymatic assays, or sometime it may be simply by looking at the SDS page and looking at the molecular weight of your protein as well, because we are talking about the cells which are overexpressing your protein. Now, you got the different fractions of the ammonium sulphate. Now, suppose you got 60% is the fraction where your protein is present. Now, I have the 60% ammonium sulphate fraction. What I will do is directly I will start running the HIC column. Okay. If you remember, we have discussed this in the past also, that why the HIC is a preferred column before the ion exchange chromatography, because now you already have the ammonium sulphate into this solution. What you can do is you can directly add some more salt which is required to have the exposure of the hydrophobic patches present on these proteins, and then you can directly run the HIC. Once you are done with the HIC, you are going to get different fractions. Okay. Now, imagine that I got a fraction where the 30% ammonium sulphate is giving me the fraction which contains my factors. Then what I will do is I will take this factor and I will do a dialysis step, and that actually is going to reduce the level of the salt present in this particular fraction, and then after that I will do a ion exchange chromatography, followed by I will do the gel filtration, and that actually is going to give me the purified protein. So, if I will be keep using the combination of hydrophobic interaction chromatography, ion exchange chromatography, gel filtration chromatography, and within the ion exchange chromatography I have the option of using the anion exchange chromatography or cation exchange chromatography, and using all these combinations probably you will be able to get the purified protein. If you follow this particular type of scheme, definitely there will be more losses of your protein from the cell, which means the recovery as well as the yield is going to be low, compared to that if you have the affinity tag, then you can directly use the affinity chromatography, and that is it. In the single step you may get the more than 90% purified protein. Once you are done with this, then you can just simply run the gel filtration chromatography, and that actually will give you almost 100% pure protein, because the gel filtration chromatography may remove some of the aggregated material from your protein, and that is going to give you the close to 100% purified protein. So, with this we would like to conclude our lecture here, and in our subsequent lecture, now we have produced the protein. In the subsequent lecture we are going to study how you can validate or how you can actually be able to characterize this isolated factor, and how you can be able to show that the factor what you are producing into these host cells is of good quality, so that you could be able to use them under the downstream biotechnology applications. So, with this we would like to conclude our lecture here. Thank you.